Good evening, everyone. My name is Minty Ledger, and I'm the Interim Executive Director for Cambridge University Health Partners. And it gives me great pleasure to welcome you to the fourth Cambridge Biomedical Campus on Virtual Tour event. Throughout this series, we have very much enjoyed beaming to you in your homes the best clinicians, researchers and academics from across Cambridge to explain their work, what drives them, and their views on how healthcare is being transformed by progress and technology. Last month, Professor Richard Gilbertson, Director of the Cancer Research UK Cambridge Centre, told us all about the impact that the new Cancer Research Hospital would have on health and care. And that's an event that you can watch on the CBC YouTube channel. Tonight, it is my pleasure to introduce Billy Boyle from Owlstone Medical. His work on breath biopsy is incredibly important. Through a miniature chemical sensor sited on a silicon chip, this technology is able to detect volatile organic compounds, substances produced or modified by metabolic processes in the body, and changes in which can be characteristic of specific disease and offer clinicians a precious means of early detection. As previous speakers in our series have discussed, early detection is our greatest opportunity to catch and treat the many forms of cancer. It's going to be fascinating to hear what Billy has to say on the potential of his work to change lives now, but also to hear what the future might bring. As usual, throughout this evening's talk, if you are watching through Zoom, you can ask questions at any time using the Q&A box, which will then be addressed at the end of the talk. We will do our best to include any comments made by those on YouTube Live, but please don't be too disappointed if yours is not included, as we do tend to have a very chatty audience. And finally, to reassure you all, you won't be seen or heard throughout this event, but it will be recorded and made available on our website afterwards, and a link will be forwarded to you by email. So, without any further delay, let me hand over to Billy and wish you all a very lovely evening. Thank you.
So good evening, everyone. I'm very happy to be talking to you tonight. Sadly, we can't do it in person, but again, very happy to be here. So what I want to try and do this evening is talk a little bit about our company, Owlstone Medical, our technology breath biopsy, what we're trying to do, but also how we got there as a business, kind of warts and all, some of the stories as we developed the technology and grew the company. At the heart of what we do is analyzing chemicals on breath, so an approach that we call breath biopsy. And our core company mission is to try and save 100,000 lives. That's what motivates me. That's what motivates our entire team when we go into work in the morning. And I think we see two big problems in healthcare, and one was touched on already. Too many people are diagnosed with disease when it's too late to cure, and often too many people are on treatments that aren't effective for them. So, that's especially true in cancer, where we know survival chances are 10 times better and treatment costs are 10 times lower if you can pick up disease sooner. So fundamentally, we're trying to develop this breath biopsy technology to address some of these challenges. And there's really three key pillars behind the business. So with the technology, we use it ourselves to try and discover biomarkers in these different clinical applications. But what we also realized is that we can make this technology available to a much broader research community. So this includes academic researchers here in Cambridge, over in the US, and also pharmaceutical customers, who are often trying to find better biomarkers to predict which patients will respond to which therapies. So a big component of what we're trying to do is to say what you can detect on breath is novel and new, but make that available to as many people as possible to conduct that research and really push the boundaries of what the technology can do. And then the last bit is looking at the data that can be generated, through our trials, but also through trials with these partners as well. And that's really where a lot of the discovery and the insights are made. The more data that effectively you can mine, the more insights and the more discoveries you can make from it. So on the front page, I showed you our team. We've got about 170 people based here in Cambridge. And we started life back in 2004 in a dark little dingy basement in the Cambridge University Engineering Department. So myself and two other researchers, David and Andrew, we were engineers by background, were interested in sensor technology, and spun Alstone out in 2004 to develop and commercialize this sensor technology. And a lot of the focus originally was on security and industrial applications. I'll give you a few examples of that. But what we decided to do 4 or 5 years ago is say, can we take the same technology and start to apply it in medical and diagnostic applications? And that's obviously a very different business, you know, so the people that we need, the types of studies that we need to do, how we commercialize, that's very different. So we decided to spin Alstone out as a separate business. And since then, we've grown to this team of 170 people. And we've been very fortunate to win the backing of investors such as Horizons Ventures, which is a venture arm of Li Ka-shing, which has done a lot of support here in Cambridge at CREK and MRC as well. So, yeah, we're very proud of the team that we have and where we've come. And I think one of the things that it's kind of tempting to do is kind of look just at that element of, you know, we've grown a team, we've raised investment, but actually along the way, there's been a huge amount of challenges and difficulties. So I want to kind of give a bit more of a background in the story to those early days and how the company has evolved over time. So here's me, Dave and Andrew. As I mentioned, we were 3 engineers who were, you know, technology geeks, interested in trying to start a business, but had no clue about business or how to commercialize technology. And I think we were really lucky because in the Cambridge area, there was a very vibrant entrepreneur community, but also a lot of mentors and support from people who had been there and done it before. So we entered business plan competitions. We were able to, you know, tap into this, you know, experience and expertise broadly around the Cambridge area. And then we went out and we said, okay, we've got an interest in technology. Let's try and raise some money. And I think, you know, thinking back to the early pitches we did, they were completely and utterly disastrous because, again, we didn't know what we didn't know. I remember being in one early pitch where the investor said, you know, what's your margin like? And I didn't know what margin was. I didn't know if it was good or bad. So I took a bit of a guess and said, it's good, hoping that was the right answer. And thankfully it was, but I think they saw through our inexperience. We didn't quite get that investment. But what we were able to do, I think, was, you know, A, learn quickly, but B, you know, again, really tap into this fantastic wealth of experience that's in the Cambridge area. And we feel very grateful that so many people have helped us so many times over the past. So the core technology that we originally developed is called FAMES. So the way to think about this, it's a bit like an electronic nose. And what makes the nose special is you can program what chemicals you want to detect just by changing the software in it. So essentially you can say, I'm interested in chemical X, use exactly the same microchip spectrometer, just change the software. And what it does is effectively pass that chemical through to be detected, and everything else that might be in the background effectively gets filtered out. So you're able to zoom in and home in on very, very specific compounds. So, you know, again, we'd come from this microchip background, and we thought, at that time, we thought our business model is going to be, you know, like Intel or ARM, you know, we'll have these chips, we'll sell millions of them. And I remember when we got our first purchase order for a device, it was like a check for $100. And yeah, that's all they wanted because they wanted to evaluate the technology. And I remember around that time, you know, we were trying to ask what our business model actually was. And again, go back to the earlier point, I don't think we quite knew what business model actually meant. But I think what we had to do was adapt quite quickly to say, you know, this isn't just about the technology, it's about how the technology actually gets applied and what are the types of real world applications that we can solve with this. So we had this core capability to sniff out chemicals at incredibly low levels with very high degrees of specificity, so you don't get very many false positives or false alarms. And when we first spun out, there was quite a lot of funding coming from the US and the UK around trying to detect toxic chemicals. So this can be things like explosives, toxic hazards, chlorine, CW agents, and those types of things. And we were fortunate here because it allowed us to focus beyond just the chip and think about an entire product that we could make, a product that could service and solve a particular problem, and use that as a way to try and win not just investments, but also contracts that would help us turn our kind of ideas into reality. So over the years from that first spin out, about $70 million went into that business, and about half of that was through investment, and the other half was through contracts that we won from the likes of the MOD and the DOD and Department of Homeland Security in the US. So that kind of got us, up that first step of technology to product. But in the last recession in 2008, 2009, government contracts dried up very, very quickly. So what we had to do then is think, well, how do we redeploy or use this technology in other types of markets and applications? So we started to look at industrial applications. So here, this is things like in food and beverage manufacture, trying to find contamination. In oil and gas industry, it might be looking for trace levels of compounds such as methanol and crude oil, for instance. So again, we're able to take the same core technology, and because you can program what you sniff on the chip, put it into instruments that we could deploy into these industrial applications. And we had a good degree of success there, and I think crucially, it allowed us to survive through those lean periods when there wasn't much government funding. And later on, once government funding came back again, we had survived as a business and we're able to kind of pick up that business in the security products. But again, at the heart of it, there's a flexibility in what you can do with this approach. And with this particular instrument here, we had researchers coming to us from the from a broad range of different academic backgrounds, but there was a lot of research in infectious disease and oncology, where people were interested in the idea of chemical markers and whether or not they can be used in diagnostic type applications. So our approach there was to say, listen, there may be, we just make the instrumentation. Why don't you take it and see what you can do with it? And I think what we noticed over time was as more and more of these academic users were integrating the technology into their clinical studies, they were collecting patient samples, they were processing those samples and then publishing those results. And we started to look at it and say, actually, there could be something here. So one of the groups that we worked very extensively with very early on was looking at the volatile chemicals that come from urine and how that can potentially be used in the diagnosis of colorectal cancer. So again, here it was pilot studies, there's not a huge amount of data here, but for us it was very, very encouraging. And that's when we really started to say, should we as a business focus a lot more on these types of chemical markers in diagnostic applications? And I touched on it earlier and during the introduction as well. One of the things that became apparent to us when we started to look at this in a bit more detail was just the scale of the problem that exists in cancer. And this, to this day, remains for me one of the most shocking statistics I've ever seen, that 1 in 2 of us are going to get cancer. So that's parents, partners, children, grandchildren, 1 or 2 will get cancer. So it touches practically everyone, either directly or through their families. And again, the thing that was really staggering when we looked at this in a bit more detail was that there was a huge variation in the potential outcomes or survival for different cancers. For some, screening is effective, or the treatments are pretty effective. For others, which are extremely deadly, they may be not as common and quite rare cancers. And then you look and you see things like colorectal cancer and lung cancer that are, some of the biggest cancer killers, affect a huge number of people. And actually, there's not been a lot of meaningful progress in survival chances. So in colon cancer, it's better than lung. It's gone from 25 to over 50% 5-year survival. But lung cancer has been barely any progress over the last 20, 30, 40 years, really. And the thing is, the answer is in sight. It's in plain sight. So we know if you pick it up sooner, survival chances are so much higher. So going from 90% at stage 1 colorectal down to 4 or 5% at stage 4. So, we were looking at, these types of problems, the technology that we have developed and evaluated by the academic customers. And then in 2012, my wife was diagnosed with colon cancer. She was diagnosed at late stage, so at stage 4. And that's when, again, it really becomes real in a way that I'd never contemplated before, how important early detection actually is. So this is us on a wedding day with our 2 kids. She had treatment for a couple of years, lots of surgery, chemotherapy, but the cancer came back and she didn't survive, and she died in 2014. And again, the story's not unusual. Almost everyone listening, almost everyone will have some type of personal connection to the absolute horror that cancer causes. And when you look at colon cancer, here we actually have screening programs. But despite that fact, it still kills 215,000 people every year and costs the economy $99 billion. And again, it is better in some respects than other cancers where it's just no testing whatsoever. And when you look at colon cancer screening, the types of approaches that get used are typically a fecal-based test or colonoscopy or some combination of both, depending on the country. And the reality is patients don't like it. They don't like to do these tests. So when you look at the compliance rates, so how many people actually show up for a test when they're eligible, the numbers are pretty low. And that's a big problem in its own right. So the nature of the test really matters. And that's why we think breath-based testing has a real role to play, because it's very acceptable from a patient perspective. And the second issue that you have here is that the tests themselves actually aren't that good. So if you had 100 people with colorectal cancer, half of them, if they don't show up because they don't want to do the test, the other half will develop late-stage disease. For those that do show up, if the test sensitivity isn't high, i.e. if it's not very good at seeing the cancer when it's present, you'll still have a high number of patients go on and develop late-stage disease. So again, here we know the problem that needs to be solved, and it's kind of easy to state the solution. You save lives by picking up the cancer sooner. But it's a very, very difficult problem to actually solve. So when we started to think about this, we had our technology, but what we also saw was that there was a lot of data and evidence out there from cancer sniffing dogs. And I'm sure a lot of people listening will have seen or heard something in the news about cancer sniffing dogs. And here, in these types of studies, they will get tumor sample or urine sample, and they will blind the dogs to those samples, so they don't know if it's a healthy sample or a cancer sample. And actually, from a statistical standpoint, these are pretty robust studies. So the dogs are picking up something. So the million-dollar question is, what is it that they're actually picking up? So when you look at these types of chemical markers, and you look on breath, the most obvious question is, what is it that you find on breath when you breathe out? And a lot of us actually know a lot more about this than we did, 9 months ago because of the COVID pandemic. One of the things that's present on breath is breath aerosol or respiratory droplets. So microscopic droplets that contain proteins, bacteria, but also viruses like coronavirus. And what you also find in breath are thousands of exhaled chemicals, what are called volatile organic compounds. And it's these chemicals that we're interested in from a diagnostic standpoint. So again, breath is great because it's non-invasive, but also you can collect a sample absolutely anywhere. So if you think about the challenge of how to get more people testing, actually being able to go to where they are and collect a sample is a big, big advantage. When you also ask the question, where do they come from? So some originate locally within the airways. That makes sense. The cells in your lungs are producing volatile organic compounds. But actually most originate from the blood, and that makes sense as well. So if you think about when we breathe, we're exchanging oxygen and CO2 from blood to the airways, and you have thousands of these VOCs that come along from that exchange from blood to the air. And that's important because it means you can think not just about respiratory diseases or diseases of the lung, but diseases which are in other locations within the body. And when we start to think about this in the application to cancer and what are the types of things that we're interested in, the analogy that we have in our head is around cancer imaging. So most people will be familiar with PET scans. So this is an imaging technique where a tracer is administered into the body, and because the tumor cells have an altered metabolism, they are hungry for this sugar, so they tend to take up more glucose. And what you can do when you image is look to see where those areas of high uptake are, and that then helps to say that there's a tumor in that particular location. And there's two conceptually important things here as it relates to what you can then potentially do on breath. So the first is, cancer does have an altered metabolism. And when you look at the vast majority of volatiles you find on breath, they are volatile metabolites. So you have the chance to look at alterations in these volatile metabolites and tie that back to alterations with the tumor metabolism itself. So they're endogenous to the body, a part of the body. The second thing it highlights is this principle of actually maybe you can target the cancer using an external probe. And here what they're doing is obviously using a probe that will release something that you can see while you image. But the question that we ask is, well, why take someone to a scanner? Can you not just design a probe that will release a unique compound on the breath should there be a tumor or cancerous cells present? So it's these two approaches combined that we think make breath biopsy quite interesting for early cancer detection applications. And so just to elaborate that on a little bit more detail, when we think about these endogenous VOCs, there's different types of processes that we try to understand, what's going on in the body, what's going on in these tumors, and therefore what are the types of volatiles that get released into the blood and ultimately into the airways. And we have a team that spent a lot of time, thinking about that core biological understanding as much about the technology itself. And then in the second approach, essentially what we're trying to do is say, what is different about a cancer cell compared to a healthy cell? So one of the things that we can exploit is to look for enzymes which are normally present within the cell, but in a tumor or tumor microenvironments sit outside the cell, and target that with a specific chemical. So if that chemical sees that enzyme, it then releases a unique compound out onto the breath. But again, crucially, you don't need to bring someone to a $20 million scanner to get the answer. So they're the two basic strategies that we try to deploy when we think about breath biopsy. And I've touched on some of the advantages of breath being non-invasive, etc. But actually, there's a subtle but very, very important advantage of breath compared to other sample types like blood or urine. So fundamentally, you have a limited sample volume when you think about collecting a blood or urine sample. You can't easily do it on demand. But what you can do with breath is with each exhalation, you have volatiles coming out with every single breath. And what you can actually do is trap, store, enrich, and pre-concentrate these chemicals over time, all just through normal tidal breathing. So that means a patient just sitting breathing normally over a period of a few minutes, you can pre-concentrate a very high number of volatiles from breath. And when you think about it, your entire circulating blood volume goes around your body once a minute. So if you sample over that type of time period, it's a bit like a window into your entire volatile metabolome of that circulating blood flow. So all of this sounds great. Which begs the question is, where are all the breath tests if all of this sounds so fantastic? And actually, because we were coming at this from the standpoint of, we developed the security and industrial business, so we had the technologies and the people in place, but we wanted to understand, what some of the basic limitations were, why people hadn't done this before. And some of it was very foundational stuff. Like if you don't get the right sample, you're not going to get the right answer, irrespective of how you analyze it. And then there was other things to do with just the design of studies. Often people were doing really small pilot studies, but they weren't taking those forward through into larger validation studies where you tested in a blinded population. So I think we identified a lot of things that we said, we need to solve all of these problems together if we want to have a shot of trying to make this a reality for patients or bring it to the clinic. So the technologies that we've developed, roughly you can break it down into how do you get a good sample, how do you analyze a sample, and then what are the biomarkers themselves. So I touched a little bit already on the analysis of chemicals, so using our microchip spectrometer. But the key here is you need to know what you're looking for. So you need to say, I'm interested in detecting chemical XYZ. And actually part of the challenge in a lot of the medical applications is you don't know what that is yet. You need to discover those things. And that was a key lesson for us, actually. We didn't quite appreciate that was the case until after we spun the business out. But before you can get to that, there's the sample collection itself. So I want to kind of just focus for a minute or so on that. So with our receiver device, this in essence allows you to collect the volatiles from your breath. So as a patient or subject breathes normally, the volatiles that come out of the breath, we watch the pressure trace on the breath, and we try to collect that very end bit of breath that's rich from the chemicals which are coming from blood out into the airways. So we have a very fast acting pump that switches on and draws the chemicals from your breath down into this breath biopsy cartridge. And inside this cartridge, there's a material which is a bit like a sponge. So the chemicals from your breath will stick to that sponge-like material and be firmly held there. And that's important because it stabilizes it, because it means you can then take that cartridge. So if you think of it a bit like if you collect a blood sample or a urine sample, you can take that sample and then send it for analysis. And this hadn't been solved, this problem before. And that's what we've been able to do using the receiver device. And then the one bit which I'll just touch on at the very end for a few seconds is originally when we were designing this, we said, yeah, but in order to reuse it across different patients, we need to be careful that you don't accidentally have these respiratory droplets going from a patient out into the base unit of the receiver device. So we had inline filter materials to stop that, very similar to what you find in N95 masks. But it turns out that as you're breathing out, this filter captures those respiratory droplets and what's contained in those respiratory droplets, which then makes you start thinking about, well, does this have a role to play in terms of collecting sample when you're doing COVID analysis? And I think, you know, again, one of the things that we found that was really, really surprising was a lot of things had been published saying that it looks like there's volatile chemicals in lung cancer and tuberculosis, asthma, you name it. But we found agreement between these papers. So paper A might say, we think it's chemical XYZ, and paper B might say, we think it's chemical ABC, and they don't match up. So again, I think that we had to approach it and saying, actually, the big problem is in discovery. You know, what are the true biomarkers here? So that meant that we had to make huge investments in our breath biopsy lab, where we kitted it out with top of the range GC mass spectrometer instruments. So these are very high-end instruments that allow you to do very comprehensive identification of the VOCs which are present on breath. So we made that investment not just for ourselves, but again, also for the broader research community who are trying to answer the same question. So you might have an academic researcher that doesn't have a lot of budget, but wants to say, is there a VOC biomarker in, you know, asthma or COPD? And they can just send samples to us, so they don't need to make, you know, these multi-million-pound investments themselves in this type of instrumentation. We've done that for them. We want to make that available to them. And then once you know what you're looking for, you're off to the races, because then you can easily tune and functionalize our sensor technology using those VOCs that you've identified in discovery. And then the last bit is, again, fundamentally, how do we think about which are the VOCs which are useful biomarkers? And the field had historically been thinking about just looking at if someone has a lung cancer, what's different about their breath profile compared to someone who's not got lung cancer. So look for the differences. So looking at these endogenous, quite small changes in concentration of these VOCs, and while they're there, the effects and the size is quite small. So you're not looking at a massive signal difference here. So this approach that we've pioneered, which again relates back to thinking about, you know, imaging and cancer PET scans, etc., what we call exogenous VOC probes, is to then say, no, we're going to put a probe into the body that will target you know, a particular piece of biology that we understand well, and then we'll release compounds under the breath. And we do that both in cancer, but also in liver disease as well. So I think, you know, our second massive stroke of good fortune, again, from being in Cambridge, was, you know, I remember the very first discussions we had with clinicians. You know, we said, okay, we've got this technology, and, you know, it can detect chemicals. We're interested to see if we can run it in studies to see if it may be useful in answering the cancer detection problem. And at CREK were amazing, because they said, you need to speak to, you know, Robert Rintoul and Bruce Ponder in Cambridge. I remember sitting with them, holding up the chip, saying, here's a technology. I've got no idea how you run a clinical trial. Can you help us? And they were fantastic. So, you know, they helped us to set up, and then became chief investigator on one of our lung cancer trials called Lucid, that's now recruited thousands of patients into that study to try and find some of those volatiles. And then we started to think, well, you know, is there scope to look at other types of tumors as well? And again, we're very fortunate to work with Rebecca Fitzgerald, who you've heard from already through the Cytosponge work. So Rebecca is the chief investigator on our PAN trial, which is looking to see other biomarkers in other tumor types as well. So I think we've had a lot of good fortune, and again, fantastic support, you know, from people who are on a common mission to try and pick up cancer sooner. And one of the results that we published quite recently was looking at this Evoke probe approach in patients that have liver disease. And here we're interested in, you know, the disease processes that lead to liver cancer. So, you know, there's very high prevalence of fatty liver disease that ultimately can lead to cirrhosis, and then that can lead through to liver cancer. And the basic approach here was to say, if certain substances go into the body, And if the liver is damaged and it can't quite metabolize those compounds anymore, can we look for those changes on breath? So one compound that we've been investigating quite closely is limonene, which is found very commonly in foodstuffs. So you'll find it in fruit juices, for instance. And limonene, when it goes in, as mentioned, it won't metabolize correctly, but it's lipophilic. So that means it wants to go into the fat in the body. So through this constant exposure over time and this inability to get rid of it, and it builds up in the fat compartment, essentially you have this increase in concentration that you should then find on breath, because it's a volatile compound that should come out on breath. So within the PAN trial, we looked at patients who were healthy, had cirrhosis, and patients with liver cancer as well. And indeed, that's exactly what we found. We found those that had these elevated levels of limonene on their breath were very different compared to the healthy controls, and it also depended on the disease severity as well. So I think that's an important finding for us. It shows that this approach of looking at not just the chemicals that are produced normally within the body, but exogenous VOC probes, this combination is likely to be really beneficial when you try to drive sensitivity and specificity of these particular tests. So those trials are ongoing. Again, we've had amazing support here in Cambridge. And again, there's no guarantees that it will work. That's the nature of science. It's the nature of the clinical trials. You do the studies and you see what the results look like. But I think there's a very good, not just technology, but biological rationale for why we think breath biopsy has a role to play in early cancer detection. And one of the things I mentioned earlier was that we also have our research services business. And this is essentially to try and say to as many people who are interested in breath, use this technology and see what you can discover with it. So this effectively means that you can take and receive a device, you can collect samples anytime, any place, and then these cartridges, these breath biopsy cartridges, can be removed and sent back to us in our breath biopsy lab. So that's what we have over in the science park on the other side of Cambridge. And in that lab, that's where we run the high-end GC mass spectrometers to try and discover these novel biomarkers for these clients. But I think, from our perspective, one of the really valuable things is that, clinicians and academics coming to us, they understand the problem that they're trying to solve better than we can. They understand, what words should be positioned within a clinical pathway, what other tests may be available and some of the deficiencies of those. And they also have access to the patient and the patient samples as well. So it's a very good partnership that allows you to identify new diagnostic problems that need to be solved, but allow you to ask that question of, well, actually, are there any chemicals or not, in a very time-efficient way. And, since we've been doing that, we've been very fortunate, again, to have some really fantastic customers and partners, including a lot of the leading pharmaceutical companies like GSK, AstraZeneca, Johnson & Johnson, but also research institutes further afield like Mayo Clinic and Cleveland Clinic. And when they use our technology or we deliver studies for them, one of the most exciting and valuable things that you get from that is obviously the data and the publications that arise from that data. So there's over 100 papers that have been published using our technology across a range of early detection precision medicine applications. And then what we try to do is to say, well, where does it look promising? Where should we put our own internal investment behind this as a way to try and make these novel discoveries to turn that into something that's actually going to be beneficial for patients. So there's been a lot of exciting work that's been published on VOCs in early cancer detection across different types of cancer. Again, I won't go into this in any particular detail, but just to show the types of work that gets published by our academic partners that are using our technology in pancreatic cancer, in esophageal cancer. So this group down in Imperial who used our receiver device. What was quite exciting about this work was that they looked at breath, but also tissue that they'd taken out of the body, and the chemicals had been produced directly by the tumor, and sampling directly from within the body as well, and finding biomarkers that look like they may be valuable from a diagnostic standpoint. And then a paper that was published just about 4 or 5 weeks ago, a group in Italy using the technology, where they've used volatiles on breath to identify patients that have colorectal cancer. So this is tremendously exciting. Again, like we always try to be, quite skeptical when things are at, that earlier stage where you don't have a lot of data, and say, well, you don't truly believe anything until you validated it in these larger independent test sets. But it is tremendously exciting to see the types of results that are coming through, both through our internal studies and the work that's coming through from our customers. So there's still a long way to go, but we have a very committed team, myself included, that want to make this a reality and bring this forward for patients. And I just wanted to finish on a slight diversion and talk a little bit about COVID, because I think it's obviously front and center of all of our minds. And actually, since the COVID pandemic started, everyone now knows how important breath is and what breath contains, because it's a transmission vector for the virus with these respiratory droplets. And again, here, what we had previously been doing was focused on the volatiles that get collected in the breath biopsy cartridge, and we didn't really care about the filter. That was just to stop contamination into the unit. But when COVID started to get going, our thinking changed, is that actually what's being trapped on these filter materials contains very valuable biological information. And we've been looking here answering different types of questions. So one question we're looking at is, from these droplets that are coming out into this mask, actually, do we think it's originating from the airways, or is it just droplets that are coming from oral cavity or the nasal cavity? Because if it's coming from there, you may as well just use a swab. But actually, there's early but very exciting data suggesting that what you get coming from these respiratory droplets is indeed different from what you would find with a swab, and originating deeper in the airways. And this is, again, very interesting when you think about COVID. And the way that we see potential application here is to think about how the samples are currently collected in COVID tests. I'm sure a few people listening have had to do a COVID test, are not very pleasant. You need to do the swab down the back of the throat, the nose. Often it's a healthcare provider that's collecting that sample. I had to take my own 2-year-old daughter over to Milton to get the test done there, and she did not enjoy being swabbed. And here, the basic concept is to say, well, if we're breathing these respiratory droplets out and the virus is contained on it, why don't we trap, store, and collect those, and then do subsequent analysis on it using the existing types of tests that work very well, things like PCR type tests. So again, we've had some of that lab-based data, clinical data showing that we can detect species originating from the deep airways. So we're running clinical study here again in Cambridge, looking to say, well, actually, could you replace a swab and just use a very simple collection device that integrates into a face mask or face covering, which you're wearing when you go to the shops or when you're out and about. And at the end of the day, in principle, you could just take that off and do standard analysis on it. So that's one area that we think breath may have a role to play when you think about COVID. And then the second one is when you think about the need for very rapid tests. So one of the problems of a lot of the tests is they take a long time, so a PCR test. And that's okay if you think about community-based testing. where, essentially you're going to stay at home. And it doesn't matter if you get the answer tomorrow because, you're stuck at home for that time period. But when you think about a lot of the areas where time is of the essence, who do you let into care homes, who can go into hospitals, when you think about airports, time is of the essence. And the basic concept here is that, people have COVID infection. They have elevated inflammatory and oxidative stress markers, which are volatile and should come out on breath. So having a test which can act like a rapid triage, so you can say, you are suspicious, you have some of these inflammatory or oxidative stress markers present on your breath. We need to peel you off and do one of the confirmatory tests with PCR, which is slower. But again, you've been able to do that first cut using a fast test. So that's the second area that we think breath biopsy may have a role to play. And again, we're able to leverage and build on a lot of the technologies that we have, with the Receiva, our FAMES technology, and our knowledge of the types of biomarkers that are present, with the inflammatory oxidative stress type biomarkers as well. So that's another study that's ongoing. Again, our hope is ultimately that the vaccine gets out there quick and there's not a need for any of this. But until then, we are working on this as well as the early cancer detection work to try and see how breath biopsy can make a difference for patients. And that's where I wanted to leave it. I want to leave a little bit of time for questions. I've also been told it's a very hard stop for Bake Off, and that cannot be broken. But I did just want to say thank you again for joining tonight. And also, you can help. One of the things that we need to do as part of our research is collect breath samples from healthy volunteers. So if anyone is interested and is curious as to what it's like to get a breath sample, spoiler alert, it's very easy. It's just breathing. But please do get in contact because you can help us understand more about what the range of different compounds are for people who are healthy, which then helps us find biomarkers in those where we're looking for a particular disease. So I'll leave it there. Just want to say a big thank you and appreciate any questions you might have.
Lovely. Thank you very much, Billy. That was a fantastic presentation. As we've had for many of the presentations so far, so that's great. We've had a number of questions, not surprisingly, have come in, some in advance and some over the course of your talk. So some of them will, there's kind of a random nature of them, but we'll go through and see what we can hopefully answer as many as we can. Because as you rightly say, Bake Off is there as almost like the clock striking midnight. So we better be aware of it. So someone simply just asked the questions, is this test likely to detect blood and lymphonic cancers?
Short answer is I don't think so.
Okay.
I think we tend to be focused more, big focus for us is on lung cancer at the moment. I think you have the release of endogenous compounds direct into the airways, and we can also target with the exogenous VOC probes as well. And there's a big need there. It's #1 cancer killer. So that's where a lot of our focus is. We do see some of the mechanisms and the things that we target in lung cancer are present in other solid tumors. So I do think it does translate into things like colorectal cancer and renal cancer. But that's where the principal focus is for us at the moment.
Okay. And how do you feel that you've managed to work with the NHS? There's a huge amount of goodwill for the NHS and it does a huge amount of work, but lots of companies similar to your position have said how difficult it is to be able to get to speak to the right people at the right time so that they can be pursuing a technology that they think might work, but actually when they talk to the clinicians, they've realized they've taken a wrong turn. And so how, I know you talked about the advantages of speaking to the right people at the right time. How else have you found your interaction with the NHS?
We were really fortunate because when we when we were coming at it from the security and the industrial business, one of the things that we needed to try and do there was secure a bit of funding just to get the ball rolling on it. And prior to us getting any investment, we actually applied for and won what's called the SBRI. So this is a small grant that actually comes from, it's the NHS saying we need to invest in new and emerging technologies around key areas of need for them. So they had a call out around early cancer detection, and we said we'll apply for that. So we won the first phase and then won the second phase, and that was great because it meant that we were able to work with clinicians, but also look at the health economics of the test as well, because actually there were so many assumptions that we made at the start that were totally wrong. So we thought, we must take the chip and embed it into the analyzer, and that must sit in a GP surgery. But actually, they're saying, no, there's no real need for a very short turnaround time. You need to pick up the cancer sooner, but it doesn't need to be real time. And actually, a better business model and something that fits easier from a regulatory standpoint is just to collect the sample and then send it to the lab for analysis. So I think there's a lot of crucial insights that we made quite early on. But actually, when we then think about the importance of saying, we had won funding from the NHS when it came to raising investment, that was critical because it validated, the need and, the technical approach that we were taking. As well. So I think that's really worked in our favor. And again, we were fortunate through the the interactions that we had here in Cambridge to find clinicians that really were interested in early detection, because that field is it's only over the last few years really, I think, has been prioritized, and it's getting a lot more kind of focus and emphasis on it. And that's been in Cambridge for a while, actually, again, with the likes of Rebecca Fitzgerald, and her interest in early detection goes right back.
Do you think given kind of Cambridge has its unique status as being the life science capital of the UK, do you think you would have been able to have achieved as much as you have in the relatively short period of time that you have if you had been anywhere else in the UK or anywhere else in the world? Has being in Cambridge helped you to be where you are now?
I think short answer is yes. I'm sure there's fantastic places elsewhere in the world, and definitely lots of places in the US. I think what I find interesting about Cambridge is you have a kind of mix of technologists and engineers and physicists, but then you also have clinicians and academic researchers as well. And there's an interesting kind of bridge between people who understand what the problems are and patient need, and some of the technology development. And then I think you also thread around a very good support network of people who have built businesses before, helped to secure investment that really give time back, and the others trying to do it as well. So I think there is a really kind of special chemistry about Cambridge that makes it easier for sure.
Great, thank you. Someone's asked, would like to know what the difference is between what Owlstone is doing and what Technion is doing. I might not be saying that correctly. And do you have any affiliations with Michael Costanza and his work?
Yeah. So we know both of those. So the guys in Technion are based out in Israel. So one of the things that we've seen that's been interesting is people have been looking at breath for quite a long time. So when you read a lot of the papers, they say Linus Pauling breathed into a mass spec in 1971. That was a reasonably long time ago. So it's like, where are all the tests? Mm. And, a lot of the approaches that people have tried have tended to try and use sensor technologies, or you introduce a chemical or breath, and the sensors themselves, they don't know the chemicals they're looking at. So they're more pattern based. So you'll get changes in some parameters, or the resistance within the sensor may change, but you don't know what the chemical is. And actually, what we believe is that when you're doing discovery work, you need to be able to say, I have patients with lung cancer, healthy controls. Here the chemicals are different, but we believe it's important to know, A, what they are, but then trace that back to the biological understanding as well. Say, well, why would they be there? Because quite often what you find is that in a lot of the small studies where you don't have many samples, but you have a lot of features or variables, often you get overfitting, which basically means you're seeing clouds in the sky and they disappear when you run the larger independent studies. So that's why, again, I think we've focused and invested very heavily in building out capability on discovery. And then once you know what you're looking for, it becomes easier just to program those chemicals into our chip. And again, I don't think people have looked across the spectrum of the different problems you need to solve at the same time, fundamentally.
Oh, that's great, thank you. There's a kind of collection of questions about kind of where the company will be in 2 years' time. What's the kind of the next stage? And someone's actually asked, they're currently working on a respiratory ward. What are the chances of we'll be able to use the technology which you're developing? Or will it be possible to have this as a home test? So are those sorts of things that you're looking at going forward?
They are. So again, I think there's our research products part of the business is again something that allows us to work with groups to help find novel biomarkers. It's a way for us to generate some revenue into the business, and actually that could be quite an interesting, significant business in its own right. But we think if we want to achieve our company mission of saving 100,000 lives, we need to get tests out into the hands of patients. And that's where our obsession is basically, how do we find, step one is you need to find genuine biomarkers and prove those are genuine biomarkers. And then once you have that, I actually think the technology is already in place there to deliver that. And I think when you look at, again, some of the challenges in testing with other sample types, blood-based, etc., people don't like it. They don't want to go and get that test done. I think breath solves a lot of those issues because it's very easy to give a breath sample. No one's afraid of it. And you can do it anytime, any place. So I think, the key is really, identifying and trusting those biomarkers for those different diseases. And the kind of risks or the likelihood of success is different for different diseases. So actually, cancer is the hardest problem. Without a doubt, it is the hardest problem. But when we look at the work that we do in respiratory disease, for instance, so where you're looking at different types of inflammations happening within the airways, actually there's quite a big signal, we think, that comes from that. And when we look at liver disease, where we are putting probes into the body that we know are metabolized by the liver, that we know will then come out on breath, that means that those types of applications are very, very likely to work. So it's about balancing the overall portfolio. Fundamentally, the early detection problem needs to be solved. And while there's no guarantee that we can do it, we are going to try our damnedest to try and be successful at doing that, because someone has to solve it. And I think one of the things that's positive over the last 3, 4, 5 years actually is this increasing focus on early cancer detection, through breath, but also liquid biopsy, novel imaging techniques as well. So my sincere hope is that you can collect the breath sample and have it look for biomarkers of different diseases as routinely in the GP surgery as you would get a blood pressure measurement.
Thank you, Billy. We're nearly at 8 o'clock. The time has flown by, so thank you again, and thank you to everybody who submitted the questions. And sorry we haven't been able to reach all of them, but we will endeavour to try and answer some of them online at a later date if we can. So thank you again, Billy. Thank you everyone turning up and watching. Just to let you know that we've got a couple more talks coming up on the 1st December, we have a new campaign that we're starting, the CBC Wellness campaign, which is looking all about kind of around mental health and ensuring people's mental state. And we're looking, we've got a man called Richard McGann who's coming to talk about resilience strategies for coping with COVID. And then on the 14th of December, it's a CBC on tour event again, and it's looking at cystic fibrosis, if I can say the words correctly. So all those events are on our website, or if you go to Eventbrite. So thanks again, Billy. Take care of yourself, everybody, and look forward to seeing you next time. And make sure you tune into the Bake Off. It's starting any second. Thanks very much.
Thanks a lot. Bye now. Cheers, everyone.